clusters is Haley Kearns. Haley, would you like to begin?
Yes, thank you. Hello, my name is Haley Kearns, and today I'll be presenting on the genomic diversity and similarity within E-type cluster bacteriophage. On the screen are some of the bacteriophage from St. Leo University that have been isolated and annotated after sequencing. And throughout this presentation, our viruses will be compared against other bacteriophages from other schools for comprehensional purposes. And from our St. Leo viruses, we have the Cephuviridae and Podoviridae type bacteriophages, Cephuviridae being the contractile long-tailed phages as seen with tiny miny, catastrophic, and store, while the Podoviridae phage are the short-tailed viruses like Kaijan and Wessick. On the screen is a Famirator map. Famirator maps are used to visualize the similarities and differences between phage genomes. The ruler-looking line on the screen represents the genome of the phage, while the colored blocks just above and just below it represent the genes. The genes on top of the ruler represent genes moving in the forward orientation, while the ones on the bottom represent genes moving in the reverse orientation. And as you can notice, there are colors scattered throughout the plot. The color purple represents high genomic similarity, while colors like yellow, orange, blue, and green represent varying levels of similarity, and the white will represent no significant similarities being found. As you can see on the top with catastrophic, there are 3 white genes moving in the reverse orientation, and these are orphams. And orphams are genes that are completely unique, completely their own. There's no one like them. And on the bottom, there is blue rugrat. You can see that some of the genes have called functions, like the minor tail protein on gene 36. And overall, phage genomes have quite a few genes that have uncalled functions, but we're slowly picking away at this as a community. The C-Phages program is a 2-semester-long course focused on isolating novel bacteriophage and analyzing the genome. The bioinformatics tools we use to do that will look at start and stop codons, finding transmembrane sites, and figuring out what the gene function could be. But the overall purpose of the program is to introduce students, freshmen and sophomore, to research and what it's like, as well as how the scientific community operates, because they'll be able to go and present at conferences. And it's really exciting and fun to see how excited the students get when they discover their own virus. They get to name it, and they get kind of attached to it. And they're all contributing to a really great cause that will be seen later on in the presentation. I will briefly go over what happens in the first semester of the C-Phages program. It starts with the students coming into the lab and bringing their soil sample, which will be cultured with broth and then filtered to get a phage filtrate. That phage filtrate will be screened against a bacteria. Our school used Microbacterium foliorum, and the DNA will be further isolated and purified in order to be sent off for sequencing at the University of Pittsburgh. And during all of this, we'll also be doing characterization tests like restriction enzyme digests and host specificity tests, as well as going to the electron microscopy lab. St. Leo University has been working with the C-Phages program for 3 years now as a part of cohort 11. We've isolated a total of 28 phage from the Tampa Bay area, and we've also annotated 7. 2 of them being phage that were adopted off of the C-Phages Genome Exchange website, and that's just where other universities post their phage that they didn't have a chance to annotate for other schools to take. We've also collaborated with the University of South Florida and annotated another 2 phage. But what has really intrigued us is that the E-cluster phage that we have, they're from a relatively small region in Florida, but we know that the microbiome around us is so vast and diverse in nature, and when we know that there's 10 to the 6 to 10 to the 7 phage particles per mil and 10 to the 31 phage particles in the entire biosphere, you know how large of a variety of viruses there are. So when you see only E-type cluster phages that we get, it's pretty cool. On this Famirator map, we are comparing all of the clusters that I will be going over in this presentation. And clusters are just groupings of phage based on their genomic similarity and their common host bacterial strain. Starting from the top, we have convict in EA1, store EB, Kaijan in EE, tiny miny in EF, Wessick in EK1, and fractured in EK2. And to clarify, fractured, the EK2, St. Louis University has not isolated or worked with EK2s before. We're just using it for comparison purposes. And we've also separated our Famirator maps into two so you have a more blown up version of it. On this slide is a dot plot comparing all the phage from the last slide. You can see with the big line in the middle, that's just the phage being compared against themselves, and it's a fully solid line, so they're completely similar. But if you look at the arrow comparing Wessick and fractured, the EK1 and EK2, you see some segments of genomic similarity, which is further clarified back in the Famirator map where you saw the purple section between them. Starting with the EB cluster, St. Leo worked with catastrophic and store, while comparing didgeridoo and stromboli to them. EB is typically around 42,000 base pairs in length for their viruses, and they code for about 70 genes. And what's really unique about the EB cluster is that it has such a large amount of genes in such a small amount of space, so it's very compact. With the EE cluster, we had Kaijan from St. Leo, and we compared them against Noble, Scamander, and Teddy Boy. The EE cluster phage are typically 17,300 base pairs in length and have around 25 genes. And what's unique about these guys is that it's a super short genome size compared to all the other E-type clusters, and some things of note are the 20 through 22 genes in the reverse orientation, as well as the variation seen in the tape measure protein on gene 12. For the EF cluster, we looked at tiny miny and compared them against Krampus, Haunter, and Aesir. The EF cluster comes out at around 56,600 base pairs and has around 84 genes. Overall, the EFs are very similar to one another, but you can see that tiny miny is very unique. Towards the beginning and end of its genome, it's completely different from its fam members. If we look towards the finish of tiny miny, there is a large grouping of orphams towards the end, which is highly unusual, and St. Leo is definitely interested in finding out what these do. In this dot plot, we compare the phage from the EF cluster. You can see with the top arrow, Haunter and Krampus nearly identical, but when you compare tiny miny against all the other EFs, there's very little similarities between them. For the EA1 subcluster, we had convict at St. Leo, and we compared them against Alex Adler, Gershwin, and Knox. The EA1s are typically 41,600 base pairs in total length and have around 63 genes. And as you can see with the large sections of purple, EA1s are highly genetically similar with only a couple variations. Moving on to the EK1 subcluster, we worked with Blue Rugrat and Wessick and compared them against Armawen and Tiny Timothy. What's special about the EK subclusters is that they have an unknown life cycle. Compared to the other phage that I've listed before, they've had a lytic phage life cycle, while these could be known as lysogenic life cycles. The EK1s are around 53,900 base pairs and have about 53 genes. As you can see going through, super similar to one another. And when we come to the EK2, once again, St. Leo has not worked with these directly before, but they'll be used in a comparison in a couple slides. They also have an unknown life cycle, and they are around 54,400 base pairs and have the 55 genes, usually. Here's it continued, highly genetically similar and pleasing to look at. And when it really gets interesting is when you compare the EK1s and EK2s. As you've probably noticed, there is a massive gene right in the middle of the EKs, and it's a 13 kb gene with no known function and no hint to what it could possibly do. And when you compare EK1s to EK1s and EK2s to EK2s, they're super genetically similar, but when you compare EK1s and EK2s, their genomic architecture is nearly identical, but the actual sequence is completely different, especially with the 13kb gene, so it makes you really wonder what this gene codes for and what it could possibly be doing for this virus. So how do we find out what this conserved genetic anomaly does? The University of South Florida has a doctor named Dr. Polins who is researching EK2s and their 13kb gene. And he's made some really amazing progress with that, and we hope to further collaborate with him on it. St. Leo University has also just been accepted into the C Genes course, which is the third part of the C-Phages program, where we will look at gene function explorations. So we can start figuring out with what the 13kb gene might possibly be, as well as the tiny-miny orphams, or just genes that they don't have a known function yet. What really makes this interesting and helpful and important for students is that we get to see what results could happen from our work. So the phage that we isolate and characterize are put into a database which could potentially be used to formulate a phage cocktail. This young lady, thank you. This young lady has cystic fibrosis with the specific drug-resistant strain Mycobacterium abscessus, and she got phage therapy as her treatment, and they used topical measures and inhalants to help her, and now she's doing amazing. So thousands of students across the country are contributing to this amazing cause and with the potential to save people's lives, and I think that's really amazing for undergraduate students, if you ask me. I'd like to thank the University of Pittsburgh, Howard Hughes Medical Institute, Dr. Graham Hatfull, the Science Education Alliance, which is the C-Phages program, and Dr. Claire Dennison, Dr. Duffy, and Professor Duncan. Thank you for your time. Are there any questions?
I'd like to ask a question, if I may, and that is, by the way, I really enjoyed your presentation. I think it was fabulous.
Thank you.
Congratulations, very well done, very professional, and very well organized.
Thank you.
And you're welcome. And I'd like to just ask you, what are you anticipating in the future? Is there the possibility that phage research might be in the future? Have you thought about that, or?
For specifically my future?
Yes, your future.
Yes, it's definitely a big passion of mine. I'm planning to get a PhD in either microbiology or virology, depending on where I want to go. So it's definitely something I want to continue with.
Oh, that's fantastic. Well, congratulations. Good for you for taking this opportunity to get this hands-on experience with the research, and especially in microbiology and virology. That's fantastic.
Yes.
Thank you. I appreciate you answering my question.
Thank you. If you're speaking, Miss Laura, I think you're muted.
Why mycobacterium?
For our school, because we use Mycobacterium, but for this it's Mycobacterium for her. But St. Leo University used Mycobacterium foliorum because it was just one of the ones the C-Phages program allowed us to use. So that was why we used that one. It was our only option.
Also, do you have any idea with the orphan proteins, like what exactly, with those proteins that don't occur elsewhere, what exactly might you find?
I mean, for the orphams, they're completely on their own, so nobody has any idea yet what they could possibly be doing, but the C Genes program that's coming to be new, that's kind of the purpose of it. They're trying to figure out what they're doing.
Thank you so much for the presentation and for the question.
Thank you.